All right, good afternoon, everybody. Before we get started on the last set of lectures for the course here, I just thought I'd say just a couple of things about the exam. So this is our last meeting before the exam, the final exam, which will be next week, obviously. There's not much more I can say, other than what I've said on previous exams. It'll be the same number of questions. It'll be 50 questions, and you will have an hour and a half to do the exam, and there will be an opportunity for you to review the exam at the end. So you'll get a Zoom invite, probably by the weekend for the proctoring half of things, and the exam's pretty much done, so I'll probably post the exam itself on Thursday or Friday. I talked to K-Back about the issues, Dr. K-Back about the issues we had related to students logging on to their Zoom sessions last time. He thinks he knows what the issue is, so obviously, that shouldn't be any issue, but I will, send a reminder to people that we're going to hopefully set up these sessions where you can, if you want to, you can log on the weekend and make sure the link works and then come back on Tuesday. So I would just encourage anybody to test the links out over the weekend for the Zoom, so we don't have the same issues we did last time with a handful of students having trouble logging in. With regard to the exam itself, remember, these are not cumulative exams, so it will only cover material since the last exam, and we've had 4 sessions. The distribution of the questions is a little bit different than the last 2 exams, in that I didn't feel that there was, nearly as much information on the parasites given this year as there will be on the viruses. So I've skewed things a little bit to the viruses. So I think we're looking at, maybe a quarter of the material. 12 or 13 questions will be on the zoonoses and rickettsial diseases. That was Dr. K-Back's lectures. Another 12, 13 questions on the yeast and fungi. So half of the exam will be, material from Dr. K-Back. And then, parasites, I think a bit fewer because there was less material. So maybe 8 or 9 questions, I think, on the parasites. And that, the difference will be made up in the viruses. So there'll be probably maybe 16 or 17 questions on the viruses. Okay. Are there any questions? Can everybody hear me? Can somebody?
Yes.
Yes, we can hear you.
Yes. All right. that's good to know. All right. So, I'll give my lectures now, but I'll stick around at the end. And if there's any more questions that you think of related to the exam, you can certainly let me know. So what we're going to do in this last session is finish up on the viruses. It's going to be I'm going to do 3 lectures, so I'm going to have 2 kind of 5-minute breaks between them, but hopefully we'll finish a little bit early. Certainly the third, the one on HIV, is quite a bit shorter than the other 2. So where we left off last week was we talked about viruses in general. And then, I told you we were going to go on and talk about specific examples, and that's what I'm going to continue today. So we talked about the family Coronaviridae, which fall into the single-stranded. These are RNA viruses. They were single-stranded, positive sense, and non-segmented. Non-segmented means, again, just to remind you, their genome is made up just of one piece of nucleic acid. So for this first lecture of the 3 today, I'm going to now talk about viruses that have a slightly different genome. These are still RNA viruses. They're still non-segmented. Okay, so one piece of nucleic acid. They're still single-stranded. but now they're in the negative sense. And I'll tell you what I mean by negative sense in a few slides. So they're a little bit different, and the replication is a little bit different. And we're going to touch on three families: the Paramyxoviridae, the Rhabdoviridae, and the Filoviridae. Oh, there we go. Sorry, it was a little frozen for a minute. Let me see if I'm good now. Okay, I'm good now. All right, so non-segmented, negative strand RNA viruses. So the Paramyxoviridae, and these, three families include within them some very important, clinically important species. So the Paramyxoviridae, that includes measles and the mumps, and we'll talk about those. Rhabdoviridae includes the rabies virus, and Filoviridae includes Ebola. So these are, as I say, negative strand RNA viruses. So what does that mean? Well, remember when we talked about positive strand RNA viruses, they look like mRNAs. So when they are packaged as positive strand, when they enter the cell, they can go directly to the ribosomes and be translated. Okay. Negative strand RNA viruses cannot do that. All right. So the strand cannot be translated. It's not recognized by the ribosome. In order to be translated, you need to make a positive strand copy. So you have to convert it from a negative strand into a positive strand. And those positive strands then can function as mRNAs, and they can then be translated. So this virus has this extra step. So how does it do that, though? How does it make those positive strand copies? There's no mammalian polymerase that can convert one type of RNA into another type of RNA. Well, what that means is that these viruses of your negative strand, you have to bring your own polymerase into the cell with you. So packaged within the capsid, within the virion, is not only the nucleic acid, but an RNA polymerase. All right, so the virus enters the cell, there's the negative strand, the polymerase is right there with it, and then that polymerase will start making positive strand copies, which then can be translated into proteins. All right, so positive strands viruses do not need a polymerase in their virions. Negative strand viruses have to bring the polymerase into the cell with them. So they have an absolute requirement for that polymerase. And that's the difference. All right, so let's begin by looking at the family Paramyxoviridae. So these are very important respiratory tract pathogens, particularly of small children, so infants and children. And these include two which we're going to talk about: the mumps virus and the measles virus. So there's others in this family that cause disease, but again, I'm just going to give these two as I think are interesting examples. Host range for Paramyxoviridae are humans and monkeys, and transmission is always inhalation, right? So you can't eat these viruses, you breathe them in the same way Coronaviridae, you breathe them in. So inhalation of aerosols. Here's what a paramyxovirus virion structure looks like. So there's the viral RNA, and this is another one of those viruses where it has a helical capsid. All right, so remember, the capsid protein is actually bound directly to the nucleic acid, so you get this kind of long hose-like feature, and that just all gets kind of packed together. And we refer to that often as the nucleocapsid. So a helical capsid is often referred to as a nucleocapsid made up of this protein. We don't call it the capsid protein. We generally call it the nucleocapsid protein. Now remember, I told you last day that any virus that has these helical, capsids has to be enveloped. They're always enveloped. So there's the envelope there. Now, in the middle here, inside the capsid, what else do we find? We find the polymerase. So remember, the virus packages its mixed polymerase and packages it in the virion so it can then transport that into the next cell it's going to infect and then replicate the genome. Now, within that lipid bilayer, again, which is provided by the host cell, are some proteins also, which are encoded by the viruses. And that's what's shown here on this next slide. So there's actually two proteins. There's one called the F protein. So there's always an F protein, and that stands for fusion. So that protein is responsible for the fusion of the viral envelope with the target cell. So this is one of those viruses that binds to the target cell, and the membrane fuses with the cellular membrane of the host cell, releasing this capsid into the cytoplasm. All right, so that's what the fusion protein is. Then we have a second protein. Now, this protein can be one of three different types. It can be what's called an HN protein. And again, it depends on the particular species that we're talking about, all right, within this family. So in some, it'll be what's called an HN protein. Some, it'll be called an H protein. Some, it'll be a G protein, right? So it's one of the three. HN stands for hemagglutinin neuraminidase. And we'll talk about that structure actually in the second hour when we get to the flu viruses. H just stands for hemagglutinin, and G just stands for glycoprotein, right? So and this protein is responsible for the virus binding to the surface of the cell. So it binds through whichever of these three proteins it has, and then the F protein allows the fusion to take place. So this is what a paramyxovirus life cycle looks like. So there's the virus fusing, its membrane is now fusing with the target cell. That's mediated, remember, by that F protein, releasing this negative strand RNA into the cytoplasm, which then becomes uncoated. And there's the polymerase, which is also brought into the cell. That polymerase will then, do two things. It'll start making these subgenomic mRNA. So it'll make positive strand copies of this negative strand genome. So it'll make, but it doesn't produce full length at this point. It's producing these little subgenomic mRNA. Remember, we talked about subgenomic mRNAs last week, right? So, and each one of these will encode a single open reading frame, which then can be translated to produce a different type of viral protein. All right, so there's, for example, is the nucleocapsid protein shown there in N. There's another structural protein down there called the matrix protein. All right, so each one of these little mRNAs, different types, each encodes a single protein. And that's what paramyxovirus genome looks like. It only has it doesn't produce polyproteins like the coronaviridae did, right? So the positive strand RNA viruses, they produce polyproteins. Negative strand RNA viruses, they don't make polyproteins, which means they don't encode proteases. They don't need a protease, right? Because they just make their proteins individually. So I'm looking for my cursor here, okay. So some of these, as I say, some of these obviously are made and translated in the cytoplasm. Some of these actually are produced, some of these subgenomic mRNAs will actually then be translated within the endoplasmic reticulum, which is shown over here. And the reason for this is these are the membrane proteins. There's F, so there's the fusion protein. There's HN. Right, so that's a hemagglutinin neuraminidase protein. So these are going to be membrane-bound proteins. So they get translated in the endoplasmic reticulum, inserted right away into membranes, and then they all get transferred out to the plasma membrane. Meanwhile, in the cytoplasm, all these other proteins are being made, which are going to be components of these new virions. In the meantime, while this is going on, the polymerase will also, start making new genomes. So there's the negative strand at the bottom. Remember, that's what's going on. So this is going to be a two-step process. You're going to make a plus stand full-length copy and then use that as a template to make more negative strands, because these are intermediates. These will eventually just be destroyed, but this is what you're trying to make more of, these negative strands to package. So as I say, this is two steps. Make the positive strand as a template, then that will be used to produce more negative strands. All right, so let's look at a couple examples of these, and we'll begin with the measles virus. So measles virus, Paramyxoviridae family, obviously. This is an acute childhood disease, one of the most contagious diseases known to mankind. It's still a major cause of child mortality in developing countries. Just to give you an idea of the mortality that can be associated with this, in 1875, this virus was introduced for the first time into Fiji. And Fiji was, at that point, a relatively immunologically naive population. The natives there had not been exposed to a lot of different, they've certainly never seen anything like the measles before. And that virus, within a year, pretty much wiped out a quarter of the population. So everybody got infected and about a quarter died. So it can be pretty serious if you're immunologically naive. In the US, if you look at 10 years before the vaccine came along, we were having about 530,000 cases per year in the United States. That led to about 50,000 hospitalizations and about 450 deaths per year. So it was pretty serious before the vaccine came along. Worldwide mortality in 1980 was about 2.5 million, and this was the point at which there was a major global effort to address the problem with measles. But at that point, in about 1980, globally, about 2.5 million deaths per year. That was substantially decreased by a program, which I'll talk about in a minute, to the point where by year 2000, it was down to about 650,000. And by 2015, we were down to about 135,000 cases per year. So there's been a major effort to vaccinate and reduce the worldwide cases and worldwide deaths. The virus serotypes only one, okay, which is great, because that means if you're vaccinated, there's only one serotype, so there's only one antibody response. This virus doesn't really mutate that well. It has a very good polymerase, and so pretty high fidelity. And so you only really have to you get vaccinated when you're young, and then you can get a booster to it, but that's it. You're probably not going to need another measles vaccine for the rest of your life. You'll have that pretty much lifelong protection. Incubation time is about 10 to 4 days. So 10 to 4 days before you start showing any symptoms. And then the symptoms start. It starts with fever, cough, right? Sore throat, runny eyes, and headaches. So that's pretty non-specific at this point. It's going to feel like you're certainly not going to be aware you've had measles unless you're aware that you were exposed to somebody. You're certainly, as I say, you're just going to feel like you have a cold. Then, okay, something called Koplik spots appear. And Koplik spots, I don't know if you can see, this is a view inside somebody's mouth. There's their tongue. And there are these pinpoint white spots on the tongue. If you see white spots on the tongue, you have to think measles. All right, particularly if the person has not been vaccinated. So white Koplik spots next, and then that's followed shortly within a few days by a rash, this diffuse, what's called a macular. Macular rash means it's raised, so it's kind of a bumpy rash. It generally starts behind the ears, all right, behind the ears, and then will rapidly spread to the whole body. Okay? Now, sometimes it will be associated with pneumonia. That's more rare, and even more serious complications. So in about 1 in 2,000 infections, we see encephalitis. Treatment, no specific treatment, but there is a vaccine. So you immunize young, the first vaccination at 12 to 15 months, and then you get a booster at 4 to 6 years. And as I say, only one serotype. So it's not like the flu where you have to get vaccinated every year. That pretty much will keep you going into late into your life. In terms of the pathogenesis of the infection, the replication, the virus infects. Remember, it's respiratory, so it begins in the respiratory epithelium, but then it will start to spread to other cell types. So it can infect monocytes and macrophages, endothelial cells, epithelial cells, and it's when it starts to infect these other cell types that the clinical symptoms appear. Eventually, it will attack the skin surfaces and it will attack your immune system. And that's why you get these rashes. When children do die, it's usually associated with a variety of measles. So as I say, sometimes it's associated with pneumonia. That can be a problem, pretty severe diarrhea. The important thing I want you to take home actually from this slide is on the left here, and this is just showing the clinical symptoms. So notice that, and this bottom part is just to show you the timeline. So this is days after infection. So here I'm showing right at the beginning of that red line there, the clinical symptoms starting at about 8 days. At this point, you're infectious, right? And you have one of these most infectious viruses in the world inside you. The problem is for the next 3 to 4 days, you don't know you have the measles. So although you're highly infectious, you think you have a cold. And it's only when the complex spots start, followed by the rash, and you're still really infectious. But at this point, you're thinking measles, okay, I can be isolated. But this child here, through those 3 or 4 days, has a bad cold. They could be at school. And if they're at school in a classroom, and that's a non-vaccinated population, every child in that classroom would get infected by that child. That's how infectious it is. All right, so what happened in 1980? The Measles Initiative, okay? And this was before widespread vaccination was taking place. Measles, about 2.5 million deaths per year. As I said, this was American Red Cross, CDC got involved, United Nations got involved, UNICEF was involved. And by 2014, 85% of the children worldwide have received that first dose of that measles vaccination before their first birthday. And it's estimated that something like, because of this program, something like 17 million children's lives have been saved by the vaccination. Okay, so that's all I wanted to say about measles. Let's talk now a little about the mumps virus. Again, a common acute disease of children. Common, I say, there is a vaccine, so common in an unvaccinated population. This generally, children tend to get infected between the ages of 5 and 9. Again, just one serotype, and that's important. There is absolutely a vaccine, MMR. That's when we get measles, mumps, and rubella. All right, so measles, we talked about. Mumps is this one. Rubella is German fever, and we're not going to talk about that in the course. Infection in this case causes inflammation and enlargement of your salivary glands or your parotid glands, right? And 35% of cases subclinical, so it's just like a bad cold. But others, the majority, will show very swollen and painful parotid glands or salivary glands. And this is what it looks like right here. So this little boy this is really, and this will be on both sides. So it's bilateral. His salivary glands are so swollen and inflamed, you can't even see his jawline anymore. And if he opens his mouth, that's going to be very, very painful. All right. And, now if he opens his mouth, you look at the back of the throat and you see nothing. Everything looks fine, because right away you might be thinking, okay, this looks a little bit like a staph. Maybe it's a staph infection. The symptoms are similar to a staph infection, but the throat generally looks pretty normal. But because the virus is actually not in the back of the throat at this point, it's down in the salivary glands. It's a systemic infection. So it's spread by viremia. So this virus actually gets into the blood, and its major targets, as I say, by this point, it's in the salivary glands. It can also get in nervous tissues, sometimes rarely, very rarely, into the testes, mammary glands, so some other places as well. But this is the distinguishing feature. Swollen jaw either side, can't see the line of the jaw, and very painful to open your mouth. Epidemiology, again, we're talking, although we get vaccinated for this in the United States, there's a lot of places in the world that don't get vaccinated. So the mumps is seen worldwide, generally between January and May. And it seems to come in cycles for reasons we don't understand. So it seems to spike every 2 to 7 years. But again, unvaccinated populations. And in an unvaccinated population, it's pretty infectious too. You're going to get 95% are going to be infected by age 15. But again, one serotype, that means you can only be infected once. You get it once, it's awful, it's painful, you get over it, and then you don't have to worry about it again. And two, it's pretty much because we have vaccinations in the United States, you don't see too many cases. But in 2016, we saw a spike. We saw about 3,000 cases. That's the highest we've seen in about 10 years. When you see these spikes, they're generally associated with the anti-vaxxer movement, which again is something that goes up and down in popularity. As the anti-vaxxer movement becomes more popular, we tend to see more cases, and then it'll go down again. Pretty easy to control, as I say, good vaccine. Again, you immunize at 12 to 15 months, then it's the MMR. Same vaccine combination that you use for measles, you also use for mumps. All right, let's move on so that's Paramyxoviridae, and I just wanted to give you those two examples. Let's move on to another family of negative single-stranded RNA virus, and that's non-segmented, and that's the Rhabdoviridae. Okay, so very similar virus to the Paramyxoviridae, but a different family. And the most important or clinically important species within this family is the rabies virus. Okay, so rabies, it's obviously causes rabies. So that's important in a lot of different animal species. It's a zoonotic transmission. It can be transmitted from animals to humans. It's also an economically important disease of fish. There are other viruses within this family that cause diseases in fish. These have an envelope, so they're envelope virus, and they have an unusual, see it in a second, elongated, a bullet-like shape. So again, that's very nice diagnostic. So see what the virus looks like there. It looks like a bullet, round, square on one end, rounded on the other end. So there's our rhabdovirus again. So looking like bullets, there it is. Here it is here. So these are enveloped. There's the capsid, and this is the again, a helical capsid. All right, so it's made up of the nucleocapsid protein. So we refer to this as a nucleocapsid surrounded by this, unusually shaped lipid membrane. There's an important protein in there. This has got a G protein or a glycoprotein. That's what it uses to bind to its target cell. And of course, this is negative strand. So what are you always going to see in there? You're going to see an RNA polymerase, right? So there's the RNA polymerase. That's with respect to this virus, referred to as the L protein coming into the cell with the virus. So rabies is an acute fatal viral inflammation of the brain, death usually through respiratory paralysis. So what happens is your lungs become paralyzed eventually. So as this spreads, it'll spread to your brain, and then it goes back to and gets into your lung. So you'll have all this we'll talk about the symptoms in a minute, all these neurological symptoms, and it'll actually come back down into your lungs and cause paralysis of the lungs. So you'll go in to the hospital. Well, you'll be in the hospital probably already. At that point, you'll go onto a ventilator. But if you're showing symptomology like that, it's terminal. So you won't survive the infection. The virus is transmitted to humans through the bites of a rabid animal, and it's also can be transmitted between animals that way as well. Kills about 50,000 people per year worldwide, which is a pretty low number. Actually, you don't see too many cases of rabies anymore, and only kills about 3 people per year in the United States. And we'll talk about this in a minute, but that is almost always associated with a scratch or a bite from a bat. So bats are the problems in the United States. That being said, it's a major problem for animals. It actually kills millions of animals in the United States every year. So it kills animals as well as humans. But we don't see too many transmissions to humans anymore. So let's look at what happens with a rabies infection.
Professor?
Yes.
Can I ask a quick question? Do you know what exactly causes the foaming at the mouth?
There is the hypersalivation, I think you're referring to. Yeah, and there's a hydrophobia as well, which is a fear and avoidance of water. And we'll get to these symptoms in a minute. But the foaming is just overactive salivary glands. So because the salivary glands themselves become infected, right? So you're producing a lot of saliva, it starts to run out, as you can see on the figure here, you see it kind of running out the mouth, okay? So it's related to the infection of the salivary glands themselves. The virus infects a lot of different tissue. Okay, so you get an initial infection here. They were seeing something that almost never happens, which is some kind of wolf or a fox. I'm not even sure what this is supposed to be. This animal is supposed to be, but it's biting this individual in the leg, right? And so it's initially it's good. It could be, as I say, a scratch or a bite from a bat. And what you're going to do is, so the virus is now in the muscle. It almost always starts in the muscle, and the virus will actually replicate in the muscle. And then after replicating in the muscle, and it can stay in the muscle for a fairly long time. Now, it's usually about 1 to 2 months, all right? But it can be shorter than a week, or it can actually be up to several years, all right? So there's a lot of variability. Typically, it would just replicate in that muscle for about 1 to 2 months, and there would be no symptoms at that point. Then, okay, it will enter the peripheral nerves. Okay, so it'll move out of the muscle into the peripheral nerve, and then it becomes a problem because then it's sequestered from your immune system. Once it gets into the nerves, the immune system can't see it anymore. From the peripheral nerves, it'll make its way into your central nervous system. And that's when you start to see the first symptoms. And this happens very quickly, peripheral nerves to central nervous system. And again, it can be very non-symptomatic at first, fever, headache, tiredness, but then this is going to progress. You're going to get neurological symptoms, anxiety, confusion, hallucinations. You may actually even go into coma at this point. Somebody mentioned the hypersalivation, this fear and avoidance of water. Nobody understands that. And then a difficulty in swallowing. And then eventually it'll get back, as I say, down into the lungs, and you will then basically, you won't be able to breathe anymore. You'll get this paralysis of your lungs. Now, once those clinical symptoms start, so once the virus leaves your muscle and enters the peripheral nervous system, the mortality is 100%. There's no recovery. Once the symptoms start, that's it. I feel like there was on the cover of a Scientific American I don't know, 3 or 4 years ago, may have been longer now, there was actually a report of one individual who showed symptomology and actually survived. That's the only person out there. I don't know if there's been an example since. And that was a massive clinical intervention that they managed to survive. But as I say, it's okay. It might be, maybe it's 99.999% death, but it's pretty high. Diagnosis is unfortunately generally post-mortem. It can progress pretty quickly, so it's easiest to diagnose after you die. There is a DFA, or direct fluorescent antibody test, to use that is pretty much 100% effective. And this is an antibody that recognizes the virus itself. So what you'll do is you'll take a section through the brain of the person who's died, and you'll use this antibody, and it will interact with the virus directly, and so it'll fluoresce. So that's a pretty good test. And everybody, if that will always be positive. In about 50% of cases, you might see these Negri bodies. Again, you're going to do a section through a brain. This is what I'm showing you here. The arrow is pointing to a Purkinje cell, and this sort of pink stain, that the tip of the arrow is touching on here. This is called a Negri body. This is actually viral nucleocapsid protein that's being produced in the cell. And it's being produced at such high amounts in the cytoplasm in the cell, and all in this one location, that it actually, you can stain it. You can actually see that, and that's very unusual for a virus to do that. And that can be used as a diagnostic again for rabies. Antemortem, so if you suspect somebody has been infected and you want to do tests, there's not one test that works. There are several tests that you need to do to confirm it. So you will use a direct fluorescent antibody test, but you'll also, and that's basically that's the last one, an antigen detection. So you're using an antibody to detect a viral protein. You'll also do an RT-PCR test. And so there's a variety of different tests that you can do, but there's not like one definitive test. You have to do several to confirm the diagnosis. Prevention and control, there's an extremely effective rabies vaccine available. No, we don't all get it because there's so few cases in the United States. Not everybody really needs it, but there are certain populations. For example, veterinarians will get the vaccine. Park rangers will get the vaccine. We used to get this vaccine from dogs. It used to be transmitted from humans, from dogs and cats, but they get the vaccine now. Dogs, if you have a dog, I'm sure your dog has a rabies vaccine. And so that's why now the spread to humans is so much more rare. And we'll take a look at some data on that in a minute. Post-exposure treatment, you can actually, this is the only virus where you can actually do a post-exposure treatment with both passive immunoglobulin and the vaccine. And the reason you can do it with the vaccine, remember, is that virus will actually stay in your muscle. So if you suspect that you have been bitten, if you've been bitten by something, by an animal, and that animal then tests positive or scratched, or you've been playing with an animal, and that animal tests positive for rabies, you can go to your doctor and you can get both immunoglobulin and they will give you the vaccine. Because if it's in your muscle, it can stay there for several months or even years before it makes it into your peripheral nerves. So that gives that vaccine time to work, right? So the vaccine can actually work on a lot of people. So you're going to do both antibodies and the vaccine. As I say, that's the only case. So if you've been, as I say, if you've been exposed and to an animal that has had rabies, that's what they're going to do. They're going to give you passive immunoglobulins and the vaccine. Once the symptoms appear, there's no real effective treatment. As I say, we've only had one individual who recovered. All right, so here's cases of, this is animal rabies in the United States between 1955 and 2001. So the yellow is total. Now you'll notice that most the majority were actually in domestic animals, right? And a lesser amount in wild animals. And most of the cases in humans, like when we get infections, was from these domesticated animals. So you get bitten by a dog, you get bitten by your cat or a cat, right? So, but once the rabies vaccine was introduced, this number went down dramatically. So now just a handful, right? Whereas, you know, the number of, again, this is just infections. This is not infections in humans. This is infections in domestic animals. Whereas the infections in wild animals continue to go up and continue to rise. So when we saw this drop, this was coupled by a substantial drop in infections of humans. So since 1960, about 90% of cases, animal cases, occur in wild animals, bats, raccoons, and skunks. Domestic animals, things like cats, cattle, and dogs, they now account for maybe less than 10% of the animal infections. These are the major terrestrial reservoirs of rabies in the United States. So this is, as I say, it's a bigger problem for animals than it is for humans. So skunks, obviously, raccoons, if you're on the East Coast, raccoons are a problem. And there's another area of skunks in the South, foxes can be infected. There's areas where coyotes can be infected as well. But overlaying all of this is bats, or, you know, these are terrestrial reservoirs. Bats are not a terrestrial reservoir, right? So they're flying, and bats are everywhere. And as I say, human infections in the US are typically associated with bats, and that can lead to 1 to 3 deaths a year. The problem with bats is, like, if you get bitten by a skunk, or if you get bitten by a raccoon or a coyote or a fox, you know it, right? You know that this is a problem. This I have been bitten. You feel it. It's a massive bite on your arm, and you're going to run off to the doctor right away. And you know, they'll say, okay, we don't know if this had rabies, but you can get the rabies vaccine and immunoglobulins right away. All right, so they'll just give you that precautionary, and you're probably going to be absolutely fine if you haven't got any symptoms. The issue with bats is you can get bitten or scratched by a bat and not know it. And that's where these 1 to 3 cases come from. So this can be a child sleeping in a bed, all right? This can be somebody working or cleaning an attic where there's bats in the attic, and a bat might fly into you or something, and it might scratch you, and you would never, if you were a child, that would not wake you up. If you were working in an attic, you would not even realize you had been scratched by that, or necessarily know that you've been scratched or bitten by that bat. So you don't even realize it. So the first time you realize you have a problem is when you have the symptoms. And of course, if you have the symptoms, that's terminal. So that's, as I say, why when we see a few deaths in the United States every year, it's almost always a case where somebody got infected with a bat and never realized that that they'd had this interaction, this bite, or this scratch. All right, so that's rabies. And now let's end this first hour looking at filoviridae. So this is the third family of negative sense, non-segmented RNA viruses. And the most clinically important member of this family is the Ebola virus. So Ebola virus, we've all heard of it, first appeared in the human population in 1976. First appeared in the Congo in the Ebola River Valley, which is where he got his name from, and as well as Sudan. And that initial outbreak ended up killing about 600 people. Very unusual virion. This is what the virion looks like. It's 2 different kind of types of staining of the same thing. It's kind of a filamentous virion. So it looks a bit like well, it's like again, like a hose pipe, right? So a twisted hose pipe. The disease is called Ebola hemorrhagic fever. It is, one of, probably next to the rabies, one of the most deadly viruses known to humans. So it has a mortality rate of about 50 to 90%. You do not want to get Ebola. There are actually 4 subtypes, 3 of which are known to cause disease in humans. Where it comes from, its natural reservoir. So it comes to humans from animals. When we have outbreaks, that's a zoonotic transmission. But pretty much everybody figures it's bats. So the best candidate, I think the one that most people would agree on, is that it originates in bats. So the reservoir is the bats. Now it can get to humans in different ways, but the virus, the Ebola virus, probably originates in fruit bats. Transmission can be an infected, and we'll talk about this a bit more in a minute, can be an infected animal to a human, or you can have a human to human contact. And transmission is almost always blood. So a blood or a bodily secretion, but almost always blood. This is not a respiratory infection. So this is where the blood of an animal gets on your skin. And if you got a cut or something on your skin, that would be your primary infection. If 2 people, if it's a transmission to human, it's going to be a blood. It's going to be a blood transmission. And that actually type of transmission is actually quite common with this kind of virus because of the symptoms itself. So the symptoms, incubation period is about 2 to 21 days. This is followed by fever, headache, muscle aches, and then diarrhea. That's very progressive, vomiting, rash, your eyes turn red, and then finally bleeding. All right, so you lose all your clotting factors. You have internal and external bleeding, so your whole skin starts to bleed. All right, and you've got internal and external bleeding, so your body becomes covered in blood. Well, of course, this is a problem for healthcare workers who are working with you, are trying to treat you, because if they get any of that, you're covered in blood, and if that blood gets on them, that can infect them. So notice these individuals here are actually, this is a dead body, and it remains infectious even after the person dies. So even if you're handling the dead body that has the blood, if you get that blood on your skin, it can infect you. So notice how these workers are completely covered in this protective clothing. There's a vaccine now. Before that vaccine came along, this is the only way you could stop an outbreak, because it was really these healthcare workers that were being infected. All right, outbreaks. As I say, the first one was in 1976, shown here. And then every, 3 or 4 years, or, even greater distances, we'd have these sort of small outbreaks in different areas of Africa. And so this is showing the different outbreaks, and this is the total number of cases. The red shows the deaths, the blue shows the number who survived. So in every outbreak, more people die than obviously survive. And that took us through to about 2014, but really, even with one of these outbreaks, you were only getting a few hundred cases, right? What happened, however, in 2004, and that's because these outbreaks were really in pretty remote villages. So what happened in 2014 is we had our first outbreak in a more suburban area, and that was in West Africa. And then we had a major, as I say, major outbreak. And so this is, as I say, starting from 2014, between 2014 and 2016, in this, what we call it a single outbreak, a West African outbreak, but it's a number of different countries involved. The actual number of cases was much, much higher than it usually was. So there's 28,000 people. of those, 15,000 were confirmed, 11,000 total deaths. But, whether you want to divide that 11,000 into this number or you want to divide it into this number, it's still a very high percentage of deaths. So the disease mechanism from this virus. So where do we get it from? Well, again, the reservoir is almost certainly bats, all right? So this virus comes from bats. We're pretty sure about that, through bats. And then it can be, and we can get infected directly from bats. But it's also thought that we might be infected by intermediate. So monkeys, for example, are a major culprit in this. They are thought to be an important vector. So the bats is the reservoir. It would infect the monkey, and then the monkey would serve as a vector and transmit it to infected humans. Now, it's not by, this is kind of a little bit misleading, this diagram here. So I told you it's really blood, and when it's humans to, animals to humans, it's the same deal. It's a blood infection. So it's not like, people eat the meat, right? So you'll eat the monkey. So people think, oh, if I eat monkey, I can get Ebola that way. That's not the way you get it. It's the people who are handling the meat, okay, or carving it or working, who get exposed to the blood from the meat. They'll get that blood on their arms or on their body, and that's how they initially get the infection. All right, so somebody, bat is bushmeat, that's eaten in different parts of Africa. Monkey is certainly eaten. So the people who are handling the bats, handling the meat, that one person will get infected, and then they will end up in hospital, and then all the people working on them, the healthcare workers, will not realize that this person has it, and they will all get, if they're exposed to the blood, they all get infected, and that's how you get these massive outbreaks. It's almost always associated with these healthcare workers who are working with these, these bodies that are covered in blood because of both the internal and the external bleeding. So once you get, infection can be through a cut or an abrasion. It can be through a contaminated syringe. All right. And here it's showing, it almost seems like you're eating it that way. That's not what's going on here. This is, again, an infection with blood. It really, nobody really thinks this, you cannot breathe this virus in. So, but if the blood gets into your nasal passage, it can be transmitted. That's that way as well. And, I think this must be an older slide, because I don't really think anybody at this point feels that, I say the blood can get down into your lungs. That's what it's showing here, into the lungs, but you're not going to get this virus by eating contaminated meat. Replication causes damage to your immune system, cytokine storm, necrosis of the liver, just a lot of things going wrong. Then you're going to get hemorrhage, because, as I say, your clotting cells become, you just basically lose all your platelets, and that's going to lead you to edema and shock and death. Again, the source, the reservoir, is almost certainly fruit bat. The fruit bat can, infect, so anybody working with fruit bats, or, collecting them and handling them to produce as a food, it's the handler, not the person eating it, the handler of the bats who's, going to get infected. That's going to be that initial case, that one individual. But fruit bats are known to infect other animals, dukers, shrews, but, primates essentially are supposed to be the main culprit, gorillas and chimpanzees. And again, whoever is handling this meat, processing this meat, they're the ones that are going to get infected. Here it says consumption of infected bushmeat. Again, this is an old slide. Nobody really thinks so much anymore that it's consumption of the meat that's the problem. It's more now what we're seeing with all these new cases. It seems to be the handling of the meat is really the problem. Prevention and treatment, obviously careful hygiene, you want to reduce exposure. But now we have a live, in 2019, a really good vaccine became available. It's a live attenuated VCV-based vaccine. And so what will happen now? So the best way now to stop an outbreak is to make sure that all the healthcare workers who are working within a hospital setting where patients are being treated, you have to make sure they're vaccinated. So again, it's like rabies. Not everybody gets vaccinated, but healthcare workers, who potentially are in locations where there might be outbreaks, they get vaccinated. So that's, that we're going to see a lot less cases of Ebola now that we have this vaccine. It's an excellent vaccine, actually. So I think we're going to see this disappear pretty soon. There's no really approved cure, but you can, treat the symptoms. So not everybody dies. If you have an aggressive treatment of the symptoms, you can certainly survive one of these infections. Okay, so that's it for the first lecture. So that's the, as I say, negative strand RNA viruses. Right now it's 2:56. Why don't we take a 5-minute break? I'll certainly answer any questions, and we'll start up again with the second part, maybe at 3:00, let's say 3:01. Okay.
Professor, for the Ebola virus, what?
Dr. Whitehead?
Yes.
I actually had a friend from New Mexico who found a dead bat in her apartment, and she actually had to go get the rabies shot, and she said it was like $4,000 that she had to pay to get.
She's lucky that she got it for $4,000. I've heard much higher numbers from some places. Yeah.
And she just found it dead on the floor of her apartment, and she found a marking on her arm, and she's like, well, this could be a bat bite, but she didn't know. She had no idea. So it was just.
Oh, that's so crazy. Interesting. Did they test the bat at all, or did she take it in and have it tested?
Yeah. So she ended up taking the bat in a bag to the ER with her. So I'm not sure what the tests came back as, but it was just an interesting little saga that she had posted. So.
Yeah, but it's not like they're not going to. I mean, it must have been scary. But it's not like they're going to wait for the results of the test, right? If you go in and you feel like a bat might have scratched or bitten you, they can go ahead and test that bat. But the first thing they need to do is give you the vaccine and give you the immunoglobulin. Did she get both the vaccine and the immunoglobulins? Do you know, or did they just give her the immunoglobulins?
I'm not sure. From what she had said, they definitely gave her the vaccine, but I'm not sure.
Yeah. But you're right. It is very expensive, crazy expensive. Yeah. All right. So I think we're recording. So let's get started. Okay. So what we're going to do in the second hour. So now we've done paramyxorhabdoamphilo. Now we're going to move on to a third type of virus. Still, we're talking about RNA viruses, but now we're going to talk, again, still single-stranded, still negative sense. So up to this point, the same as the pararhabdoamphilo, but now they are segmented. So this family that we're going to talk about are segmented viruses, which means that their genome is made up of more than one piece of nucleic acid. And every piece of the nucleic acid is distinct. So it's like taking their total genome and cutting it into pieces. So these are not copies of one another. They're distinct pieces of nucleic acid. And the most interesting member of the most interesting family that fall into this grouping is the orthomyxoviridae, which includes the flu viruses. So that's what I'm going to talk about for this next, hopefully less than an hour. So flu viruses, major cause of respiratory illnesses worldwide and certainly in the United States. So if we look in the United States, the most common cause of respiratory illnesses are rhinoviruses. So they cause common colds. They account for about a third. Influenza is second. They account for another third. And then the others, we talked, remember, about coronaviridae last day. They probably come in, maybe at about 20-25%, I think we were saying. And then the other small groupings are things like parainfluenza, respiratory syncytial, and the adenoviruses. So influenza, now this was before SARS-CoV-2 came along. So, by next year, by the time I get this lecture next year, we'll have a better sense of, the total numbers. So I suspect that coronaviridae is going to move up to certainly up to number 2, if not number 1 next year, but we don't have the numbers to update yet. So anybody comes to you and says, oh, I had a terrible weekend, I had the flu this weekend, but I feel better today, is lying to you. They did not have the flu this weekend. Most people there are most people who said they had the flu did not have the flu. Okay. They had a really bad cold. So what's the difference between flu and a cold? Well, a cold is an upper respiratory tract infection. So the virus will get in your nasal cavity, the pharynx, maybe down into the larynx. So you can feel pretty rotten for a couple of days, but it'll generally resolve in less than a week. The flu, okay, gets into your lungs. All right. So it engages both the upper and the lower respiratory tract. So the virus can spread down into the lungs. So it'll come in through your respiratory secretions. Even though it gets into your lungs, it's a localized infection. So it never gets into your blood. It just spreads through the respiratory tract. Okay. But unlike a cold, it involves the entire respiratory tract. And the difference is, as I say, a cold might last for a couple of days and you can feel really lousy. A flu is going to last anywhere from 7 to 10 days, the symptomology, maybe even longer. You're going to be in bed and you're going to feel like you are dying or you're going to want to die. And anybody out there who's listening who's really had the flu, I think you know what I'm talking about. So we're talking about fever, chills, so even though there's no viremia, even though it's in your respiratory tract, it feels like every muscle in your body is infected. All right. So you have muscle soreness, cough, headache, fatigue, can have vomiting and diarrhea, which is something obviously you don't have with a cold. And it hits children especially hard. So the people at the highest risk, there's 3 high risk categories. Children themselves, and they have a high morbidity, which means essentially they are going to show most severe disease, especially if they're under 1 year of age. And the problem with young children is if an adult gets a flu, they're only actually infectious themselves for a few days. When children get the flu, they can actually shed the virus for weeks. So even when they're feeling better and all healed up and back at school, they can still infect other people or other children. So it spreads much more rapidly in children than it does in adults. And the vaccine doesn't actually work as well in children as it does. It's a pretty good vaccine, but it has a lower efficacy in children. So flu that's why children are a high risk group. Other high-risk groups, adults with underlying health problems. This virus gets into your lungs. Obviously, if you're a smoker, if you have some kind of pulmonary disease, anything to do with your lungs, but also diabetes, if you're pregnant, this can also, it can put you in a higher risk category, not just for getting the flu, but having a bad outcome. And the elderly, which is where we have our highest mortality, about 20,000 deaths per year in the United States. So if we look at this graph here, the problem with the flu is this purple bar. So the yellow bar is your clinical symptoms, right? So they start here at about 4 days after you've been infected, you start to show all this symptomology. And here, this is showing going out to, maybe 9, 10 days. Often it goes longer than that. So you can be pretty sick for a week or even longer. But then you start to get better. Your symptoms go away. But this shows the pathological changes in your respiratory tract. So even though you feel better, there's still a problem in your respiratory tract, which makes you extremely prone to secondary infections, all right? And this is particularly problematic in the elderly, okay? The respiratory tract, they feel better, but the tract isn't normal yet. So the type of complications we see, the most common one in the elderly is pneumonia. Now, pneumonia, that's excessive inflammation, airway occlusion, right? So this is a problem, particularly in the elderly, but what's causing the pneumonia? Bacterial infections, strep pneumoniae, influenzae, Staph aureus. All right, so we've heard about these way back at the beginning of the course. These are part of your normal microbiome, but now your respiratory tract is so is abnormal, and it allows these bacterial species to now infect more readily, and that happens commonly among the elderly. So you see a lot of pneumonia associated with flu in the elderly because of this. And as I say, this is this long window of opportunity for these bacteria to infect. It can also, if you get the flu, it can exacerbate other health problems that you might have. If you have asthma, it can make your asthma worse. Cystic fibrosis, it's a problem. Congestive heart failure. So all of these, if you have these conditions will be made worse by the issue with the virus infecting your lungs. So these are just additional complications. Okay, so let's look at the influenza viruses themselves. So as I say, Orthomyxoviridae families, we have different species. We have type A influenza virus. We have type B and type C. Type C, we're not even going to talk about. It's a very mild disease. It's like a cold. You don't worry about it. Seldom even gets diagnosed. Type A, by far the most common virus that causes flu. So this is type A influenza. Actually mutates at a very high frequency. So there are actually many subtypes, as you'll see within this, within this species. And these subtypes are determined by two segments, the H and the N segments. Just put that in the back of your brain. We're going to get to this in a minute. Okay, so these are the two most important segments, the H segment and the N segment. All right, so many subtypes. Type B influenza virus, all right, only one subtype, but there are many variants within the subtype. So this will have many subtypes, and in each of these subtypes, there'll be a lot of different variants. Here we have one subtype, but many different variants. Already you're thinking, well, so many subtypes, so many variants. Now you should be already thinking, okay, this is why we have to get a new flu vaccine every year, which is true. And we will get to that. Disease associated with these infections, we have seasonal flu. These are yearly epidemics. Usually in the US, they peak in February. So it's from November to April is flu season, peaking in February. These are due to what we call antigenic drift. And I'll tell you what that's all about in a few minutes. And then, of course, we have these pandemic flus every 10 to 40 years, and that's associated with antigenic shift. And again, this is something that antigenic drift is something that a lot of viruses can do. Antigenic shift is something that's unique to the segmented viruses. So here is the viral structure. So these actually these viruses actually have an icosahedral capsid. So there's the capsid. So instead of, up till now, everything we've been looking at has these helical capsids, but influenza has a icosahedral. So it's like this hollow ball. And inside this hollow ball is the genome. And remember, these are negative single-stranded RNAs, but the genome is divided up into 8 different segments. Each segment encodes pretty much for a single gene, and each has a different name. Remember, I told you about the H. I'm going to keep these 2 in mind. There's the H segment. There's the N segment. The H segment encodes a gene for the protein hemagglutinin, which is a protein that's expressed on these are enveloped virus. So that's hemagglutinin. It's inserted into the envelope. So that's produced by the virus. And the N is neuraminidase, okay, which is a second viral protein that's expressed on the surface. And that's where the tremendous genomic diversity in the virus comes from these 2 segments, the H and the N. What that means is they mutate, everything mutates, but most of these segments can't tolerate a mutation. You put a mutation in here, the virus becomes inactivated, but you can put lots of mutations in H and N, and that's why they are so variable and why we have so many subtypes. And again, we'll get to that again in a few minutes. So segmented minus single-stranded RNA producing 1 protein per segment. There's another segment, I call that PA, because these are negative. Remember, these are negative sense virus. All negative sense viruses need to bring their polymerase into the cell with them. So the PA, that's the segment that encodes for the polymerase. Another protein you should be aware of, this M2 protein, it's an ion channel that's found, again, it's encoded by the virus. It passes through the virus into the inside. This will actually can regulate the pH, okay, inside the virus. So it's a pH regulator, and we'll get to that a little bit later. We'll get back to it, why that's important. Influenza life cycle. So here's the virus attaches to the surface of the cell. And this virus, even though it's enveloped, that does not fuse directly with the plasma membrane, it comes into the membrane in the endosome and then gets released by the endosome into the cytoplasm so that it can be uncoded. And so the virus is released, the nucleic acids, the polymerase is released, all in the cytoplasm. But look what happens next. Everything then goes to the nucleus, and replication takes place in the nucleus. Now, I've told you before that all RNA viruses replicate in the cytoplasm. There's one very important exception to that, and that's flu viruses. Flu viruses, for whatever reason, we're not going to get into it today, actually replicate in the nucleus. All right, so they go to the nucleus. There's your viral RNA, that's negative strand. It'll make positive strand RNA copies, which will have to go back into the cytoplasm to be translated to make proteins. And then, of course, it'll, like all other negative strand RNA viruses, it has to go through this 2-step, it's negative strand, it goes to a positive strand. This we call the cRNA, and then back to the negative strand. This is how you make genomic copies, new genomic copies for packaging. We discussed this back when we looked at the non-segmented negative strand viruses. All right, attachment, okay. So the receptor for this particular virus, all right, the receptor on the target cell is sialic acid. All right, so sialic acid is a molecule that's linked to galactose, and it's found on host membrane proteins. So if a cell expresses these proteins in its membrane, and those happen to be linked to a sialic acid, then the virus can bind to it. And the protein that the virus has that binds to that sialic acid is the hemagglutinin. So hemagglutinin binds to the receptor by binding, shown here, to the sialic acid. But remember, So there's a lot of different proteins involved here. Just if you have a protein and many proteins get sialic acids attached to them, if the protein has a sialic acid attached to it, then the virus can bind to it, again, through this hemagglutinin protein. All right. Okay, so that binding between the hemagglutinin and the sialic acid on the epithelial cell surface. So one place you're going to find a lot of sialic acid is on epithelial cells, which is why this virus infects epithelial cells so well. And that's necessary that interaction is necessary for both binding and infection. But the sialic acid creates a problem. And here's the problem here. So the virus infects the cell, okay, it replicates, and then it buds off the surface of the cell, right? So the virus makes its hemagglutinin, it makes its neuraminidase, it cleaves off the surface of the cell looking fine, but as it cleaves off, it's picking up a cell membrane. But cell membranes tend to be filled, okay, particularly epithelial cells, with sialic acid. They just have a lot of sialic acid on the surface. So as the virus comes off the surface of the cell, it has proteins stuck in it from the host cell that still have the sialic acid on them. Now, why is that a problem? It's a problem actually for a lot of different reasons, okay? The first reason is if this virus, now this new virus that's been made, has sialic acids on its surface, it can now act as a receptor for other virus. So the virus all starts to clump together. So this virus interacts with another virus. So we've got one with hemagglutinin, the other with sialic acid. So now these viruses start treating one another like their target cells. So all the virus clumps together. So that's a problem for the virus. What else is a problem? Well, mucus. Remember, this is a respiratory virus. To infect you, our respiratory cells are covered with mucus. Mucus actually contains a lot of sialic acid in the mucus itself. So if this virus is trying to make its way through through the mucus to get to the underlying cells, it gets stuck. It gets bound to that sialic acid. So that's another problem created. But probably the biggest problem, okay, relates to viral budding. So as that virus releases or buds off the surface of the cell, okay, it cannot bud off the surface of the cell if there's sialic acid present, okay, right? So if there's sialic acid present as the virus tries to bud off, that sialic acid will actually prevent the virus from budding. So sialic acid is creating crazy problems for this virus. So how does the virus get around it? It gets around it by having this other protein, neuraminidase. What neuraminidase does is it cleaves sialic acid. So as the virus goes through the mucus and it makes its way to the target cells, the neuraminidase will cleave all of that sialic acid that's in the mucus, and it will clear this path from the virus. As the virus, encounters other virus, right? So, how is it going to prevent itself from clumping? It prevents itself from clumping by the neuraminidase will actually clear or cleave all the sialic acid off the surface of itself. So it actually cleans up its own membrane, right? So now the virus can't, as I say, aggregate with other virus. So it prevents that problem. But the main thing that neuraminidase does, that it absolutely has to do, is it allows the virus, those are sort of indirect effects, but the real direct role it plays here in virulence is it allows the virus to bud. Okay, so if that neuraminidase is inactive or not present, it directly prevents that virus from budding off the cell. So the virus cannot disseminate. So it can get in, it replicates, but it can't get out without the neuraminidase. Okay, so 3 different functions for that extremely important viral protein. So influenza virulence factors, the hemagglutinin, important protein on the surface, binds to the receptor. The neuraminidase cleaves sialic acid, helps the virus in a lot of different ways, but the most important one is it allows the virus to bud off the surface of the cell. So it allows the virus to disseminate. The M2 ion channel is also important. Remember I mentioned this earlier. This is also provided by the virus, all right? This actually allows viral uncoating. So remember how this virus comes into the cell. It comes into the cell in the endosome, and then it needs to get from the endosome into the cytoplasm so that it can uncoat. And in order to do that, when the virus enters the endosome, it needs to change its pH because the pH of the endosome is really, really low. So this ion channel then allows these ions to flow into the virus. The pH of the virus now changes so that the capsid can now be released into the cytoplasm and uncoated. And so this can only happen if you have this ion channel present, all right? This will, again, all come important when we talk about treatments for influenza a little bit later. All right, now let's talk about some terms I've mentioned already. Viral variation, huge problem with influenza. That's why we have to have vaccines every year. That's why we always seem to be dealing with these periodic outbreaks or periodic pandemics. All right, so there's two mechanisms by which this virus can undergo variation. One is antigenic drift, the other is antigenic shift. Antigenic drift is gradual change by mutation. And so this is where the virus just accumulates mutations, and flu viruses do accumulate mutations very rapidly for a reason I'll talk about in a minute. So for example, these different strands, the H strand, for example, if it mutates at a very high frequency. I'm not showing in the N strand here. It also mutates at a very high frequency. So there's tremendous variability between those two strands, the H strand and the N strand. And that's all caused by this random mutation that this virus undergoes. Okay, I say a lot of viruses have bad polymerases and they undergo a lot this mutation. And that's what we call antigenic drift, when the virus changes because of mutation. Antigenic shift is a more abrupt change, and it's caused by a reassortment. And I'll talk a lot more about this in a few minutes, but this is something that is unique to the segmented viruses. So we'll get back to it. But let's talk about the antigenic drift first. So what causes antigenic drift? It's caused by error-prone polymerases, all right? So a polymerase needs to copy RNA, all right? In this case, it's an RNA virus. That polymerase that comes into the cell with the virus, it needs to copy the RNA, but every polymerase makes errors, right? And different polymerase has different error frequencies. So, you make the errors, and then usually a polymerase will have a proofreading function. So it'll then go back and correct its errors. What happens with many RNA viruses and their polymerase, they don't have those proofreading functions. So they make the errors and they don't function and they don't fix them. And so they are very error prone. An influenza virus, their polymerase is very error prone. So they have a very high mutation frequency. So they go through error prone replication. And as I say, if you put a mutation into your polymerase, so the polymerase doesn't work anymore, that's not going to be selected for. That virus will just die. But there are two segments, the H segment and the N segment. If you put mutations into those, which are these two proteins, the hemagglutinin on the surface, the neuraminidase on the surface, the virus can tolerate those kinds of mutations. And there's tremendous variability in those two segments. And that's where most of this antigenic drift I'm talking about takes place. So we just take a look at the H protein, for example. This is the molecular structure of the H protein. So here's the viral envelope, and we have what's called the stalk, This is the hemagglutinin protein, the stalk. It's stuck in the viral envelope. And outside, we have these 5 different A, B, C, D, E globular heads, and they create this receptor binding site. That's where the sialic acid binds, right? Well, this is highly immunogenic. So this, because it's on the surface of the virus, when we get vaccinated, all right, we raise antibodies that target this, okay, this head. And there are actually, these A, B, C, D, E represent 5 major antibody binding sites. The problem is, from year to year, these keep mutating. So every year they mutate, which means the antibodies we got from our vaccination last year no longer can bind. So this virus is continually escaping neutralization by last year's antibodies. So that's why we have to have a new vaccine every year based on what the virus has most recently mutated to. Okay, so that's why we have these yearly epidemics. That's due to antigenic drift. That's why we have a flu season every year, and every year we have to get a different vaccine. So if you look at mutation rates, okay, so RNA viruses tend to be terrible. Their polymerases are terrible. If you compare a DNA virus, it makes 1 in every 10 to 8th to 1 in every 10 to 11th nucleotide, right? That's a mutation rate. That's as good as a human cell. Right, so that they have really good polymerases. RNA viruses can be pretty bad. So anywhere from 1 in 1,000 to 1 in 10,000 nucleotides incorporated. So that's a problem. So the RNA viruses, as I say, mutate more. And this is even made worse in the negative strand RNA viruses, because remember, the virus comes into the cell as a negative strand, and in order to replicate itself, it has to go through 2 steps. So it makes what's called this cRNA intermediate, and then it uses that as a template to make more viral RNA. So it replicates, every time the virus replicates, it replicates once, and then it replicates again. That's all in the same cell. So it has 2 opportunities to introduce mutations. So RNA viruses typically, as I say, mutate a lot higher frequency than the DNA viruses. DNA viruses have good proofreading functions. RNA viruses tend to be pretty bad. Right, now, because of this, what happens is, I say, that you get these segments that can actually evolve very rapidly. And the ones that are evolve most rapidly are the ones that are on the surface of the protein. And that's the surface of the virus. And that's a problem, because that's what we're raising our antibody response to, right? That's what the vaccines are working on. So the hemagglutinin is the worst. So it evolves incredibly fast from year to year, so fast that there are actually 16 different H subtypes now found in nature, all right? And by a subtype is defined is a variance of greater than 20% of amino acids, all right? So between 2 subtypes to be defined, you have to have 20% different amino acids than another subtype, all right? That defines you as a subtype. So there are 16 actually different subtypes of the H, just the H segment alone in the influenza A virus, okay? And even within a subtype, you can have variation, all right? You can have up to 15% variation within a subtype. That's why we have what's called variants. Remember, influenza type B has only 1 subtype. Okay, so it doesn't mutate at the same frequency as type A does, but it does mutate to the point where you can have a lot of variance again within that H segment. So these viruses are constantly are mutating and are under immune selection. So that's antigenic drift, and that's one way that this flu viruses vary. But another way is called antigenic shift. And antigenic shift is a reassortment of the segmented RNA viruses during a mixed infection. And this almost always involves another animal. So flu viruses, for example, can infect humans, we know that, but they also infect pigs at a high frequency, and they can infect waterfowl. It's like there's just all 16 subtypes as you'll see, of influenza type A are actually found in waterfowl. And so what will happen is, in this circumstance, you'll get these are mixed infections. And what I mean by that is an infection by two different species. So you'll have a cell that's infected by, say, we'll call the human flu virus. So that's its 8 segments. And let's say the gray is a bird flu virus. So both will infect the same cell. Now, because they're both in the same cell, what comes out can be, there can be all kinds of different variations on what went in, but what is most likely to come out is something that is a mix of both the bird and the human segments. Okay, and if this then goes on to infect humans, we have, what we call a bird flu or a swine flu or something like that. So that exchange of segments can take something, for example, that does not normally infect humans and now allow it to infect humans. So we got something brand new now infecting humans. This typically takes place when this occurs, reassortment, it typically takes place in pigs. So what will happen is, now pigs, right, can be infected by bird flu, and they can be infected by human flu. So what would happen is you'll get a pig, and there's swine flu itself. So they have their own flu as well. So pigs are basically susceptible to anything. And so what will happen is you'll get a pig that gets infected by a human flu and gets infected with a bird flu. And so now within that pig, you'll get cells that are infected by both bird and human. And so then you'll have this new, combination, we would probably call it a bird flu type flu that comes out. It's created in the pig. Now that pig will then turn around and infect a human. So they'll push it back into the human. And that's very commonly what happens. And it happens every 10 to 40 years. And that's what leads to these pandemics. And that's what antigenic shift is all about. So again, what I just said, we have a pig, all right? So pigs have flu viruses in them. So flu viruses can replicate in pigs. If a pig gets infected with a human influenza virus with its 8 segments, that same pig gets infected with a bird virus with its 8 segments, okay? So now they're both replicating in this pig. Let's say a cell within that pig gets infected by this virus and this virus. We now get this reassortment take place, and we get a new virus that's got human segments and bird segments, and it's just incubating in this pig, right? And now that pig can then turn around and infect a human with that virus. So we have this new reassorted virus now infecting back into humans, and now we have a problem. Now we have potentially a pandemic on our hands. So, waterfowl, birds are the worst, all right? So birds are the natural reservoirs for all influenza viruses. So there are actually, as I think I told you, 16 different subtypes of the H1 segment listed here. H1 down to 8, all 9, 10, all the way down to 16. So there's 16 different subtypes of the hemagglutinin segment, right? And every one of those you can find in birds. You can see waterfowl in every one of these. Only 3, H1, 2, and 3, are found in humans. There are 9 N subtypes. That's the neuraminidase, all right? So again, all 9 of those are found in birds. So that flu viruses do wonderfully well in birds, and that's where a lot of the diversity in these words, most of the subtypes came from birds. So the humans and pigs are kind of the same, right? Pigs can be infected by N1 and N2 and H1 and H3, okay? And so when we look about what's in the human population right now, all right, like what's floating around, what do we get every year? We get there's 2 different subtypes of virus that we're being infected with. And the one's shown in blue, H1N1, all right? And that flu came to us, it was called in 1977. So that was introduced into the human population in 1977. It was called the Russian flu. So there's a lot of that around. And the other thing that's around is an H3N2. Okay, H3N2 actually came early and came into the human population in a pandemic in 1968, okay, from Hong Kong. So that's what's, as I say, floating around right now. Every year when we get vaccinated against this H1N1, and we get vaccinated against this H3N2. And as I say, but several years from now, these might not be around anymore. It might be something completely different. And the reason for that is because every time one of these things happens, or many times one of these viruses breaks the species barrier, often it takes over from the existing strain. And so that's, as I say, and that's what happened here. In 1977, this H1N1 came in and took whatever the existing strain was in the human population at that time. This one took over. And then, the H3N2, when it came over, it probably outcompeted something else, another flu strain, and pushed that out. So every time one of these shifts take place, we get this change, and then we're going to need obviously new vaccines created. 1918, we've all heard about the 1918 influenza, the Spanish flu. That was a pandemic. That again, that was actually an H1N1. It's not the H1N1 that's with us now. It was another H1N1. Remember, there's different variants even within the H1. So this was a different H1N1. 1918. It actually again was an infection that went through a pig, ended up in humans, and hit about 20 to 40 million, killed about 20 to 40 million people worldwide. 675,000 people in the US. So when these things cross the species, sometimes they can be very, very serious. Here's the one from 1968, H3N2. That's the one that's still around. That was the Hong Kong one. Not showing this diagram doesn't show the one from the Russian flu, which is also around now. And so, what does it mean? Like so these things come over all the time. Well, not all the time, relatively frequently we get this breaking through the species, but what needs to happen for it to actually be officially called a new flu pandemic? Well, historically, there's 4 things that have to occur before we designate this as a new pandemic. Once, obviously, this has to be a virus coming from another species that breaks the species barrier. So it's something that's coming from pigs, it's something maybe that's coming from birds, all right, into humans. So that's one thing. 2, we have to be able to detect it. That's almost obvious, right? It has to be a new virus subtype, never seen before in humans, but now is detectable, right? So we can't hypothesize that it exists. We actually have to be able to see it, this new virus existing. The virus has to be highly pathogenic. I mean, if it comes over to humans and it actually doesn't cause any disease, maybe it just causes a mild respiratory infection, we might not even recognize that it exists, okay? Especially generally, it needs to be more highly pathogenic. That's what's already present at the time. And the virus has to be efficiently transmitted between humans. So we can go from a pig to a human directly, but if then it can't go from human to human very efficiently, you're not going to get a pandemic. The whole thing might affect, a few hundred people in a localized area at most, okay? So in order to be defined as a pandemic flu, you have to hit all 4 of these different points. So let's take a look at 2 recent, incidents where we've had flu viruses break the species barrier, one in 2003 and one in 2005. And using these criteria, let's see whether we can define these as pandemics or not. So one was, I don't remember if you remember back in 2003, H5N1, which was the avian flu, okay? And then there was an H1N1, a different H1N1, a swine flu, that hit the human population in 2005, and they're still present. So the question is, are these pandemics? Well, let's take a look. So let's start with H5N1, the avian flu that came in 2003. So this was a bird flu. So H5N1 originated in waterfowl. And it turns out that this virus, this variant, or sorry, this subtype, although humans can be infected directly from birds, it does not spread very well between humans. All right. So the only way we could get this virus is if we were, spending a lot of our time working around waterfowl that were infected with the virus. So the virus would these viruses, the birds would be breathing it out. Humans working with the waterfowl would breathe the virus in, bird to human. But when that human got that virus, okay, they then couldn't turn around and infect another human. This human right here would also have to be infected directly from the birds. Now, if you didn't get infected, you had a very high probability of dying. This was highly pathogenic, all right? So now we have a virus that is cross-species. It's coming from birds to humans. We detected it. We knew it was new. We called it the avian flu, H5N1. Had a very high pathogenicity. It was killing people, but the transmission, human transmission, was absolutely negligible. And so that meant that the only people dying were, it was localized to Asia in these markets where you had a lot of waterfowl in these markets. People were getting infected in these markets, but really only several hundred people ever got infected and died from the virus. All right. And so a lot of this waterfowl just had to be destroyed to get rid of this virus. So although we had a cross-species transmission, this did not qualify, or at this point has not qualified as a pandemic. And so, but that's not to say that this will not mutate within in a human and become a pandemic. It just hasn't at this point. Now compare that to 2005, the swine flu. I don't know if you remember the swine flu back in 2005, H1N1. Okay, in this case, the pathogenicity, okay, this again was something it turned out to be a triple reassortment virus. So it came from pigs to humans. So we got it from pigs, but it actually contains human, avian, and swine subunits. So obviously a pig got infected with a human and a bird flu, then all three viruses got into a cell, and what came out was segments from all three. Pathogenicity was about equal to, but less than, the seasonal flu that was present already in the population, okay, which is essentially another H1N1. Transmission human to human was actually pretty efficient for this one. So for example, in May 2010, we estimated that there was about 630, by May 2010, we estimated that there was about 635,000 cases, 155 countries, 13 months, mortality rate, about a total of 18,000 deaths, about 2.8%. So it was transmitting actually pretty well, but its pathogenicity was equal to, if not a little bit less than, what was already present within the population. So this one, you would probably say, yes, it probably meets all the criteria. This was a pandemic. The only thing with it, it was it actually, because it really couldn't compete with what was already present, it essentially just kind of drifted away. So there's still a little bit out there in the population, but within a few years, probably it's going to be just outcompeted by those two strains I told you about that are more prevalent. So it was a pandemic, just not a particularly serious one. So it had lower pathogenicity, but still highly transmissible. Right? So interesting. Flu treatment. There's three types of drugs on the market we use to treat the flu. Remember that M2? All right, so that M2 is your ion pump, or that controls the an ion channel that controls the pH of the virus. And so the virus, when it gets into the endosome, needs to change its pH in order to uncoat. Well, the very first flu treatments, amantadine and rimantadine, these may be familiar terms to you. These were drugs used to treat bad cases of flu. So these, for the longest time, were the only drugs available. So what they would do is they targeted that channel, that M2 channel, preventing the virus. So the virus could get into a cell, but it couldn't uncoat. And so they were actually really good drugs, very effective. But the problem is the current viruses that are in the population are both actually resistant. At the moment, they're resistant to amantadine and rimantadine. So they develop resistance within the M2 pump. So they don't work as well as they used to do. Now, as we go forward into the future, that pump may mutate again, and these drugs may become useful again. But I mean, they are still used. They're just not as good as they used to be. There are three. The main new target is the neuraminidase. All right. So the neuraminidase, remember, its most direct function is egress. So that allows the virus to bud off the surface of the cell. And the nice thing about it is it's expressed on the surface of the cell. So if you can on the surface of the virus. So if you can target the neuraminidase, it gets nice access right to the virus as it's trying to bud off the surface. And it will actually, if you inhibit that neuraminidase, all right, so this is a neuraminidase inhibitor. So I say, if you target that neuraminidase so it can't function, then the virus will start to bud off the surface of the cell, but it can't, all right? So that's what these neuraminidase inhibitors do. So it's sort of this enzymatic activity on the surface of the virus. You target it, and you prevent the virus from budding. That's its main function, but then it also has these secondary functions of the virus does manage to get off the surface of the cell. Those viruses are going to clump together because, again, the neuraminidase is going to prevent the sialic acid from, the inhibitor is going to prevent the sialic acid from being cleaved. So that's what Tamiflu does, all right? So that's why when it came out, it was a new and very effective treatment. There's a lot of effort going into developing more neuraminidase inhibitors at this point in time. So, the virus mutates. So you've got to keep ahead of it. And there's Xofluza is actually a drug that targets the polymerase. Remember, I called the polymerase PA. It's expressed on the PA. So that's a third target, and that works pretty well as well. But I would say most money right now is going into these neuraminidase inhibitors. Vaccines, obviously, we get vaccinated every year. There are two vaccines. There's what we call an inactivated split vaccine. Okay, and I'll tell you what split means in a second. And this is composed of two influenza A subtypes. Remember that I said the two most common ones, H1N1, H3N2 right now. So these are part of the vaccine. And generally we haven't talked about influenza B at all, but generally you're going to include influenza B as well. One or two variants. There's only one subtype of influenza B, but there are a number of variants. So usually, again, depending what's around, you might include one or two variants. So what you do is you grow these viruses up to mix them together, grow them up to very high titer, and then you strip the membranes off them. That's called splitting the vaccine. You strip off and isolate the membranes, which contain the hemagglutinin and the neuraminidase protein, right? And that's what the vaccine is. So vaccine is just those surface proteins, and that's what you're going to get injected with, and it works pretty well. So that's what, and again, it changes every year, and we'll talk about that in the next slide. There's a second vaccine. This is a live attenuated vaccine, cold adapted. This was an intranasal mist. So some of you may have had this in some years, although you're probably, I've never had it because I'm in the wrong age bracket. But this was actually a version of the virus. So the virus was alive, but it could only grow under cold temperatures. So they would inject this into your nose, and then it would replicate in the nose where the temperatures are fairly low. But if it's tried to spread into your lungs, it couldn't replicate in the lungs because the temperature was too hot. So that's what I mean by cold adapted. So it would just stay in the nose and you'd get your immune response. All right. And it worked pretty good for a few years, but it was only used for healthy individuals, say between the age of 5 and 49, high risk individuals, small children, and those elderly. This is the beta vaccine, and this is the one they were given. But this one, at least you don't have to give a shot, right? So people kind of liked it. This doesn't really work that well anymore. It worked really good. I don't even know if they give this anymore. It seems like about 2-3 years ago, they stopped offering it. So I don't think it works particularly well against the strains that are out there now. Again, it's something that might come back in the future, but for the moment, most of what we're getting is this inactivated vaccine. How good are these? Well, generally anywhere from 30 to 85% efficacious, right? So in any given year, the vaccine could be really great, in which case we have 85%, or it could be lousy 30%. So why not 100%? How come we can't get higher than that? And the reason we can't get higher than that is because of virus production, all right? So it takes anywhere from somewhere between 6 and 9 months to produce this virus, okay? So what'll happen is, okay, so right now, probably they're already thinking about next year's vaccine, all right? So we're getting to the end of the flu season. So now we're about 9 months away, 6 to 9 months before the start of the next flu season. So what's going to happen right now is, world surveillance will be taking place. So around the world, different labs will be isolating the influenza viruses that are out there, and they're going to see what's out there, like what has last year's virus mutated to. Okay, so what does it become? And they're going to use those new variants, okay, which are not the same as last year, because as I say, they've already gone through a year of mutation, and they're going to use those new variants, okay, to grow up to make next year's vaccine. So this year's vaccine is always based on last year's vaccine. And, so then they go through this process of strain selection. This thing is grown up in what we call embryonated eggs. So you have to grow them in chicken eggs to get them to high titer virus concentration. Then you purify the surface protein. This whole process from start to finish before you can distribute the vaccine, as I say, takes anywhere from 6 to 9 months. The problem is, while you're doing this, the virus is out in nature, is continuing to mutate. If it doesn't mutate very much in that 6 to 9 months, maybe you'll get a vaccine that gives you 85% protection. If it mutates a lot from last year, then the vaccine could be pretty crappy. I remember I think it was 2-3 years ago, there was one that was only like 35-40% effective because the virus that mutated so much. So the only way available to us really to make those vaccines more effective than they are now is to figure out a way to shorten this production. All right, so we have to get it. If we can get this from 6 or 9 months down to maybe 1 month or 2 months production, then we can start our surveillance much closer to the beginning of the flu season. And then, so that's, there's a lot of interest in figuring out how to do flu vaccine production. But as I say, that's the only way that you can really get around this problem right now of the flu vaccines not being all that effective from year to year. Okay, so that's the end of part 2. So are there any questions?
Professor?
Yeah.
Is it possible you can go over what you meant by split vaccine again? I didn't quite understand.
Yes, so split vaccine just means that, so you grow up the virus, right? So you make this mix, you've got H1N1, H3N2, and you've got your type B variant. So you grow all this virus up, all right? So it's whole virus, okay, to high titer. And then, once you have enough, you strip off the surface envelope. So you split the virus into two components. You've got your capsids, okay, and you've got your envelope. And the envelope contains the surface proteins, right? Like the H protein and the N protein. You throw the capsid away, and you're going to just take those surface proteins, and that's going to be your vaccine. That's what we refer to as split the vaccine, or it really should be split the virus. We take the virus and split it into two components, and we just use the one component, the surface component, as the vaccine. Does that make any sense?
Right. So it's the surface proteins that will end up being the vaccine.
Always. Yep. It's actually always the H protein, pretty much. A little bit the N protein, but it's always the H protein that we raise our antibody response to.
Okay, thank you.
Okay. Okay, so it's 4 o'clock. Why don't we again take a short break? Let's say 5 minutes. Everybody stretch their legs, and we will come back and finish the course. All right. All right. All right, that's not letting me advance, so let's try calling it up again. There we go. Okay, so let's get back to our little chart here. So the last type of virus I'm going to talk about, or the last infection in this course, is Retroviridae, family Retroviridae, which contains HIV. And this, again, has an even different type of genome than we've discussed so far. These viruses we refer to as the RNA DNA viruses. And so what do I mean by that? Well, viruses in this family, are able, or of this type, okay, are able at different points in their life cycle to be able to convert their genomes from RNA viruses convert themselves from RNA to DNA, or from DNA to RNA. So they can go back and forth between being RNA and DNA viruses. Some enter the cell as DNA viruses, some species, some families, and then convert themselves into RNA viruses. Some, like the Retroviridae, which I'm going to talk about today, they're going to enter the cell as positive strand RNA viruses, which we've seen already. But then once they get into the cell, they'll convert themselves into a double-stranded DNA. And then before they leave the cell, they will convert back into a positive strand RNA. All right, so that's why we don't consider them to be RNA viruses. The reason they're able to do this, so Retroviridae, as I say, HIV falls into this category. The reason they are able to do this is because they can do a process called reverse transcription, which uses a viral enzyme called the reverse transcriptase. And this enzyme does is it converts single-stranded, positive-stranded RNA into double-stranded DNA. So this is something the human genome, we don't do in mammalian cells, but some viruses can do this. Retroviridae can do it. The reason these viruses convert themselves into double-strand DNA when they get into the cell is because they need to integrate. So these are the only viruses in which their genome needs to stably integrate into the host genome. And that's part of their normal life cycle. If they don't integrate, they cannot replicate. It's part of their replication cycle. And finally, very high rates of evolution possible. The reason for that is, well, you should know by now. When I say high rates of evolution, what I mean is this virus mutates at a very high frequency. And the reason it mutates at a very high frequency, because this viral enzyme, reverse transcriptase, does not have a particularly good proofreading function. So like many of the other RNA viruses. All right, so family Retroviridae, this includes the lentiviruses. Okay, so the lentivirus is a genus within the Retroviridae family. And this genus includes the human immunodeficiency virus, or HIV. This causes, so that's where it falls. These lentiviruses, they infect humans, but there are lentiviruses that infect other animals, horses, for example. They cause chronic progressive diseases. They're unusual, the lentiviruses, in that they can replicate in non-dividing cells. Okay? Most viruses can only replicate in a dividing cell for a variety of different reasons, a cell that's undergoing division. But lentiviruses can enter into a cell that's just a quiescent cell, infect it, and start replicating. They do tend to kill cells, not all cells, as you'll see with HIV, but they do have a tendency to kill cells on viral exit, and they are enveloped viruses. Okay? Other members of the Retroviridae that cause disease in humans, there's what we call the HTLV group. We're not going to talk about them today, but these are a group of Retroviridae that are associated with human tumors. So they're related to HIV, but in a different genus. All right, so HIV is the virus, and the disease, of course, is AIDS. Just some brief history here. AIDS was first recognized as a discrete clinical entity back in 1981. It probably was around before then, but this was the first time that there was a big enough cluster in one place in San Francisco that it was recognized as something new that potentially looked infectious. Then 1983, the first retrovirus was infected from the lymph node of a patient with AIDS. In 1984, a different group isolated a retrovirus from the blood cells of different AIDS patients. They both sequenced their viruses. They found the virus they both gave the virus a different name, but when they sequenced their viruses, they found it was exactly the same virus. So in 1986, the International Committee on the Taxonomy of Viruses, or ICTV, suggested that people use the name human immunodeficiency virus, or HIV, which is what the virus has become, the species has become known as. Nothing on this slide you need to memorize. This is just, again, to give you an idea or sense of the extent of the issue that's still present with regard to this virus. So this is 2013. Actually, the data has been pretty static for 2013, isn't really moving up or down too much. So the total number of people living with living with HIV, not AIDS, those are two different things. So the total number of people living with HIV infections is about 37 million. And the majority of those cases, about 70% of them, are in sub-Saharan Africa. And then, the rest of cases are spread throughout the world, West and Central Europe, North America, about 2.5 million cases. So we account of about 7% of the infections worldwide. Let's look at the HIV virion structure. Actually, it's a little more complicated than some of the viruses we've looked at so far. Let's start with the genome itself. So the genome actually has two identical copies of these positive strand RNA. But even though it's two pieces, so two copies, this is not considered to be a segmented virus. So these are non-segmented. Remember, segmented viruses, each piece is different from one another and encodes different genes. This is a situation where this virus is simply diploid. It has two copies of exactly the same mRNA. All right, so two positive strand RNAs. Also found within the capsid are actually 3 enzymes. Now, this figure is only showing one of them. That's the reverse transcriptase. I talked about the reverse transcriptase already, right? So the virus comes into the cell and converts those positive strand RNAs into a double strand DNA. So it needs an enzyme to do that for it. So it needs to bring the reverse transcriptase into the cell with it. Now, there are 2 other enzymes that are going to be found in the capsid. That's the integrase and the protease. So I'll talk about what their function is in a minute. Then we actually have 2 capsids. So there's 2 protein coats. There's one here, an inner one that's associated with the nucleic acid very tightly. We just refer to that as the nuclear, because there's 2 capsids, we call the inner one the nuclear capsid, and then there's an outer one, which is just the capsid. All right, so capsid, nucleocapsid, made up of 2 different, each one made up of a different protein. They're enveloped, and there are viral contributions to the envelope as shown here. And those viral contributions, those glycoproteins, allow the virus to attach to the target cell. So target and attachment, again, is a little different than we've seen so far because it requires 2 receptors. So there's what we call the primary receptor. So the primary receptor is the CD4 receptor. So the CD4 is present on the surface of the target cell. Where you're going to find this receptor is primarily on T cells and macrophages. So there are CD4 positive T cells. If any of you are immunologists, you know what those are. Those are T helper cells. Okay, so that's CD4 positive T cells, as well as the macrophages. So these are both obviously cells in the immune system. So this virus infects our immune system. But the one receptor is not enough. So once it binds to CD4, it needs to engage a second receptor, and we refer to that as a co-receptor. And depending on which substrain of virus we're talking about, HIV has a lot of different substrains. That co-receptor can be one of 2 types. It can be either CCR5, that's by far the most common in North America. We see mostly CCR5 strains, but sometimes you see it will use CXCR4. Okay, so it needs, the most common, as I say, combination is CD4, CCR5, but some strains can also infect cells that are CD4, CXCR4 expressors. It's interesting because there are actually 1% of Caucasians worldwide, okay, about 1%, are homozygous for a polymorphism called delta 32. And what delta 32 is an in-frame deletion within this co-receptor, CCR5. So we call it a polymorphism, not a mutation, because the receptor works perfectly fine for whatever the normal receptor, does. I mean, it's a normal cellular receptor, so it works just fine. But the thing is, if you have this polymorphism, if you're homozygous, HIV cannot recognize this receptor, CCR5. 5. So it cannot use this as a co-receptor. What that means is that, you know, if you are homozygous for this, you're not completely, but you're almost completely resistant to an HIV infection. All right, so let's take a look at the life cycle a little bit so we can understand what this reverse transcriptase is doing. So the virus, you know, infects the cell, it releases, so it binds to the receptor, it fuses and releases its RNA, which gets uncoated from its, you know, all those capsids. And now we end up with these actually 2 copies of these positive strand RNA in the cytoplasm. We also, remember, have the reverse transcriptase, which came into the cell as well. And that reverse transcriptase in the cytoplasm will convert those 2 copies of positive strand RNA into a single double-stranded DNA, all right? So you need 2 copies to convert it into a double-strand DNA. So although this comes into the cell as a positive strand RNA virus, and that looks like an mRNA, just like coronaviridae, which we talked about last week, okay? These are never translated. So they do not go to the ribosome to be translated to produce proteins like coronaviridae and picornaviridae and all these other positive strand RNA viruses, you know, we talked about. These positive strands are immediately converted into this double-stranded DNA. This double-stranded DNA is then transported to the nucleus where it becomes stably integrated into the genome. It becomes part of the genome. This is mediated by the integrase, okay? Remember, there's 3 enzymes in the virus that come into the cell. The reverse transcriptase, the integrase, so now that's why we need the integrase as well. So that's the second one. So now we're integrated. That virus will start making mRNA, so transcribing mRNAs, right? 5' to 3'. These are positive strand mRNAs, which can then be released back into the cytoplasm. And the genome actually encodes polyproteins. So these polyproteins need to be translated to produce these proteins. But because they're polyproteins, and we'll look at this more in the next slide, they need to be cleaved. All right, so those need to be cleaved. And that's why the virus brings the protease as well. It has that protease available to then help cleave these polyproteins that are made. Those positive strand RNAs that are going out, basically the virus is now converting itself back into an mRNA, right? A positive strand RNA. They can serve as genomes for the new cells as they're made. All right, so reverse transcriptase, the integrase, and then the protease to cleave these polyproteins. So just again, comes into the cell as RNA, 2 positive strand RNA copies. I'm only showing 1 here, but there's 2 that come into the cell. They, you know, are then converted into a double-stranded DNA. And as that conversion takes place, that RNA is actually completely degraded. So once this reaction is over, all that RNA, those 2 RNA strands are gone. All right, and you're left with this double-strand DNA copy, goes to the nucleus and gets integrated. And if you look at the genome here, you'll see what I've labeled as GAG, POL, and ENV. That's 3 long open reading frames. Each one encodes a different polyprotein, which can then be, you know, you're going to make mRNAs for each of these polyproteins. You're going to make those proteins, but then these need to be further cleaved by the protease. So for example, there's GAG, POL, and ENV. You'll make an mRNA here, okay, that just encodes GAG, that encodes PEG. There's the mRNA that comes out. If you look at GAG, it actually produces 1 long polyproteins that can be translated, but it's actually, there's a matrix, that's a structural protein. There's the capsid protein. There's the nucleocapsid protein. There's some other little structural proteins as well. So each one of these then needs to be cleaved off, and that's why you need this protease in the cell. Okay, and so the protease, it turns out, it's nice. It's in the cell. It's required for these protein it's required for protein maturation. So it became a very, very nice target for drug therapy for HIV. Some of you may still remember the class of protease inhibitors that came out. All right, let's talk about the disease itself. So that's the virus. Let's talk about an HIV infection. An infection progresses through 3 stages. Now, what I'm telling you here, these 3 stages is what happens if you are not treated, okay? If you're treated, most of the time you'll just stay in this first stage. So this what happens in an infection if you're not treated. So it starts out with what we call the early stage. And this will begin about, a couple of weeks after the initial infection. You'll start to show symptoms. Now, a few people are asymptomatic. Others will very much have flu-like symptoms, right? And remember I told you, flu is bad. So you're going to feel really, really sick. So if you get this, you get these symptoms, you probably will go to the doctor. And, he'll take a history, and based on your behavior, you might actually order an HIV test and you may get diagnosed. Now, if you leave this untreated, what happens is after about 6 to 8 weeks of this early stage, you actually start to feel a lot better. You get better when you enter what we call the clinical latency stage. And that's a misnomer. I don't know why they call this the latency stage, but because it implies that the virus is not doing anything. And that's absolutely not true. The virus is still active and still replicating. But what's happened here is you have now mounted a major antiviral response. Your immune system has been activated. You are fighting the infection, and you're doing a pretty good job. So as the virus makes more virus, you're attacking and clearing those virus-infected cells. So lots of virus still around, but it's reproducing very slowly because it just, as I say, every time it infects a new cell, your host immune response kills that cell and prevents it from reproducing. And this can go on for a long, long time. This battle between the virus and your immune response can go on for up to 10 years, again, if not treated. But it invariably will lead to the 3rd stage, which is full-blown AIDS. And generally, this is associated with opportunistic infections. What's happened here is you've lost the battle because the virus is attacking your immune system. Remember, it's attacking CD4 positive T cells, it's attacking macrophages, and so it's impairing your immune system. So over time, eventually, your immune system just is gone, and then you become very prone to all kinds of exotic opportunistic infections, and that's what really full-blown AIDS is all about. And most people who die of AIDS die from these opportunistic secondary infections. And so the CDC has a clinical definition of AIDS. It's anybody who's infected with HIV whose CD4 positive T cell count is now under 200 cells per microliter of blood. Now, a healthy adult usually has about 1,000, but remember, this virus is infecting the cells and it's killing these cells. HIV kills CD4 positive T cells. It doesn't kill the macrophages, but it kills CD4 positive T cells. So over time, your CD4 positive T cells is slowly going down and down and down and down. And as it goes down and down, your immune response gets worse and worse and worse. And eventually you drop below this threshold. And when you do, basically the CDC says you don't have an immune response left anymore. You are now in full-blown AIDS. Now, this is also the time you're going to be sensitive to all of these opportunistic infections. And there's actually about 26 different clinical conditions, most of them associated with opportunistic infections that will now occur when your CD4 positive T cell count. So they're also worked into the definition of AIDS. So let's say, your CD4 count is 250, okay, but you now have Kaposi sarcoma, or candidiasis because you've had one of these opportunistic infections. Even though you're still above 200, the fact that you have one of these other 26 clinical conditions, this is just some of them, they would say you have AIDS. All right, so it's either that or one of these conditions. Pathology, what's going on here? What's causing the disease? Well, it's tissue dysfunction because of the infected cells. So what's happening is the virus will infect, as I say, primarily two cells. Your macrophages, and the macrophages actually do fine when they're infected. It doesn't kill them. But when it infects CD4 positive T cells, what it does is the virus doesn't kill the cell when it buds off of these cells. So the virus doesn't directly kill the cells. What the virus does is it triggers apoptosis. So these CD4 positive T cells that are infected with the virus, they'll make some virus, they'll secrete it, but then apoptosis will get triggered and the cell will simply die. All right, so it undergoes kind of it's not a murder, it's a suicide. Okay, so that's important to make that distinction. And you've actually, you're going to actually, those are virus infected cells, so you will kill those infected cells yourself. So your host immune response, your CD8 positive killer cells, will recognize those cells as virus infected. So those cells are being killed in two different ways. One, by apoptosis, because the virus is in them, and two, by other cells of your immune response. Okay, but as all these cells get killed, of course, as I say, you become immunocompromised. Your CD4 positive T cell goes down. If you lose your T cells, your CD4 cells, you lose cellular immunity, you lose humoral immunity. These are both arms of your immune system. And then that's what makes you susceptible to these secondary infections. So, this virus has a tremendous effect, negative impact or effect on your immune system. Obviously, as I've just said, you deplete your CD4 positive T cells, by, infected cells being cleared, apoptosis triggered as the virus replicates. But, all kinds of strange stuff are going on in your immune system. Those infected cells can actually not only trigger apoptosis of themselves, but they will actually trigger apoptosis of uninfected cells as well. So if you know your immunology, you know that CD4 positive T cells, one of their functions in the immune system is to induce CD8 positive T cells, your killer cells. If you lose your CD4 cells, you lose your CD8 cells. So those cells go down as well. And we also see, monocytes, macrophages, all the other cells of the immune system, for reasons that are not well understood, also become functionally impaired. So this is an HIV infection is an all-out full assault on your immune system. And it can take a while, but eventually your immune system will be gone. Treatment, a couple of, these is just some principles when you're considering an HIV treatment. HIV infection is always harmful. So if you don't treat it, it will, you will eventually get AIDS. All right. You don't survive an HIV infection, typically, or very rarely, unless you have some kind of treatment. It can be 8, 10, 15, 20 years maybe, but eventually your immune system will be so immunocompromised. So you have to always treat it that way. So as soon as you determine that you have an HIV infection, you have to start treating it. You're not going to wait until the person gets AIDS or wait to see if the person gets AIDS. They're going to get AIDS, so you've got to treat as early. If you treat as early as possible, you're going to have a better outcome. What you're trying to do, the purpose of treatment is to keep the viral load as low as possible. By viral load, I mean how much HIV RNA there is in your plasma, how much virus is in your blood. That's what you're trying to keep low, because that is an indication of the magnitude of viral replication. If you can keep that number low, that you're going to have a very good clinical outcome. You'll also measure your CD4 positive counts. That's a measure of the damage that's going on in the immune system. So typically you're going to keep an eye on that. Monotherapy is generally a waste of time. We learned that early in the epidemic and, through some hard lessons. And that's just because, again, a property associated with the reverse transcriptase. It mutates at such a high frequency that if you just use one drug, all you do is select for drug-resistant strains inside the individual. It's almost inevitable that the monotherapy will fail. So what we use is antiretroviral therapy, or ART, which is a combinatorial therapy. There are actually 5 different classes of HIV drugs, and the antiretroviral therapy generally consists of 3 different drugs taken from at least 2 different drug classes. So you couldn't take all your 3 drugs from one drug class. You could have maybe 1 drug from each of 3, or you could have 2 from one and 1 from another. All right, so that's where we stand right now. It's generally 3 drugs. Again, that being said, very recently a 2-drug regime was approved by the FDA. So we might see ourselves in the future going to 2 drugs. But the vast majority of treatments still that are being used by most clinics involve 3 medications or 3 different drugs. So here's your drug classes. And if you look at them, you really realize they make absolute sense. You've got your non-nucleoside reverse transcriptase inhibitors. What's the target? The reverse transcriptase, right? The viral enzyme. And this is non-nucleoside. We've talked about this kind of drug before. These just directly target the polymerase. So they bind to the polymerase, in this case, the reverse transcriptase, and prevent it from happening. The nucleoside or nucleotide reverse transcriptase inhibitors, okay, so they look like nucleosides or nucleotides. They get incorporated by the virus into these new chains, and they cause immediate chain termination. Okay, so again, they're inhibiting the reverse transcriptase, but in a different way. Now, they are considered to be 2 different drug classes. So you could take, 2 of these and 1 of these and put them together even though the target's the same, their mechanism of action is quite different. The protease inhibitors, obviously, remember the virus brings a protease into the cell with it. You can disable those proteases. The trouble with the protease inhibitors is they have really bad side effects, most of them. And so it used to be they were like central to almost all different, it used to be there was always a protease inhibitor as one of the 3 drugs. Now we see them used very little anymore. Okay, and I'll tell you why in a second. So they're still around, they're still used now and then, but they're not as effective or as important as they used to be. Entry inhibitors, there are fusion and binding inhibitors. These are just coming onto the market very, very frequently, and they're finding their way into these therapies. But here's the most important one right down here, the integrase inhibitors. Obviously, what do they target? The integrase. So they disable the integrase. And, again, a very new class of drugs. Maybe 10 years ago, we started to see these. Wonderful drugs, very effective, very few side effects. Resistance is unusual, is much rarer. And so now we're at the point where they're almost always included in ART. So, when I say, 2, 3 different drugs, one of them is almost always an integrase. So it's now an integrase inhibitor plus 2 others. Used to be a protease inhibitor plus 2 others. But now, say the protease inhibitors has been replaced by the integrase inhibitors. Okay, so remember that. An integrase inhibitor plus 2 other drugs. And usually it's, 1 of these and 1 of these, or 2 of these or 2 of these. Again, different clinics have their preferences. And that's it, right? I think we're done. Great. Okay. Any questions? All right, let's start with, are there any questions about the lecture?
Yes, Professor, I have a question.
Sure.
Can you go over again how budding is a virulence factor?
Okay, so which virus are we talking about now? Are we talking about the influenza virus?
I think so. It was where we're talking about budding.
Yes, we're talking about, okay, so we're talking about the neuraminidase, right? And so this is when we were talking about the influenza virus, and we were talking about, and I had a list of different virulence factors. Right. Okay, I remember the slide.
Oh, yeah, slide 38.
Right. So one of them was the neuraminidase. So the importance of the neuraminidase, right? I told you its most important function is to allow the virus to bud off the surface of the cell. Okay. So if there's sialic acid at the surface of the cell, a new virus is trying to bud off, okay, the virus cannot bud off the surface. And so that's why the virus needs to have that neuraminidase present to cleave all of that sialic acid and allow the virus to then bud. So it's an important virulence factor for the virus in that if the virus doesn't have the neuraminidase, it can't bud off the surface of the cell. So it's important for, and the virus, if it can't bud, it can't disseminate. And so the virus can't spread. So that's why we consider it to be a virulence factor. And, but it also makes a very nice target because we have these, these new drugs, these neuraminidase inhibitors are very, very effective because they actually target that activity and they prevent the virus from budding off the cell. So that's what I kind of meant. That's what I meant of it as a virulence factor. If the virus doesn't have it, the virus can't replicate. Well, it can replicate in the cell, but it can't leave the cell. So the virus gets stuck in the cell. So it's an important virulence factor for the virus itself, but it makes a nice drug target.
Okay, so it's important because it wouldn't allow the virus to replicate since it can't get out of the cell to infect other cells.
Well, it's careful how we use. Let's be careful now. When we talk about viral replication, we talk about that as something the virus is doing inside the cell, replicating its genome. All right. But yes, so it would, but the virus would replicate its genome, make new capsids, go to bud off the surface of the cell. And if it doesn't have neuraminidase, it's stuck in the cell. It can't bud.
Okay. Thank you.
All right. All right, so that's okay. So, please, if you have any questions, feel free, reach out to me. Any final questions about the exam next week? As I say, by the weekend, you will have, I'll have the exam posted and you'll, probably either on Friday or Saturday, like last time, you will get an invitation to a Zoom meeting. And, please, go ahead and try out that link to the Zoom meeting, right away. As soon as you get it, make sure it works. And they should be active. It depends how the different proctors, I'm going to tell them just not to put any limitation on when people can access that meeting. So you can get a chance to access it ahead of time, just to make sure, as I say, the link works. So. And hopefully we won't run into that issue that we had with the last exam. So.
Professor Whitehead?
Yeah.
I have one more question concerning the influenza lecture before you go.
The HIV lecture?
Influenza.
Influenza, all right.
Concerning the treatment slide that you had.
Yeah.
Is it just saying that those specific drugs target those proteins, basically?
Yes, exactly. So there's different drug classes. It's like HIV, right? So there's some drugs target the neuraminidase, some drugs target the that M2 ion pump, and now we have this new drug that targets the polymerase. So yeah, 3 different classes of drugs. So I just gave you the 3 different targets and the drugs that fall into those different classes, types.
Okay. Got it.
Okay.
Thank you.
Okay, well, great. Thank you very much. It's a pleasure. It's too bad I didn't, this is the second year we've had to do this course predominantly online, and I don't get the same opportunities to meet the students that I used to have in the past, and and talk about some of this stuff more. But again, thank you so much, and I wish everybody good luck in the exam next week, and good luck in your what I'm sure will be very successful clinical careers for many of you in a whole variety of different medical professions. So, all right. So I hope you all enjoy, or some of you at least, and enjoyed the content and enjoyed the course.
Thank you, sir.
All right. We'll see everybody
Thank
next
you.
week.
Thank you so much.
Thank you.
All right.
Good day.